Hello, and welcome to the Brannigan Lab Group, part of the Center for Computational Integrative Biology at Rutgers. Today we will be looking at one of our group's main projects, the effects of the thyroid hormone known as T3 on the GABA-A receptor. Let's first discuss the GABA-A receptor. GABA receptors are a group of receptors that respond to the primary inhibitory neurotransmitter known as gamma-aminobutyric acid, or GABA. GABA-A receptors are the main type of GABA receptors and are part of the cis-loop ligand-gated ion channel superfamily. Now that may seem like a very fancy title for the receptors, but let's break it down to make it much simpler. Each type of these receptors are composed of 5 protein subunits. Every one of these subunits contain a unique loop between 2 cysteine residues, which is where the first part of the name comes from. These 5 protein subunits then arrange around a pore in what is referred to as a pentameric arrangement. You may notice the color coding present in each subunit. Some mutants come in various forms designated by a Greek letter and a number. Combinations of different subunits are what create variation in function of different types of GABA-A receptors. Viewing the receptor from the top, we can clearly see 3 different subunits used for our receptor. Gray indicates the alpha-1 subunit, green for beta-1, and blue for gamma-2. The choice and the arrangement of these subunits come from previous research done on this common GABA-A receptor. Looking at the pore, we can then see where the other part of its name comes from, ligand-gated ion channel. The leucines, shown in pink, are thought to form the primary gate of the GABA-A receptor. When GABA binds to the receptor, the gate becomes more stable than the open state, so ions can pass through the pore. The ion in most cases for the GABA receptor is chloride. Now that we have a better understanding of the GABA receptor, let's talk about T3. T3 is short for triiodothyronine, named so for the 3 iodine atoms that are connected to it. T3 is a hormone that is produced by the thyroid gland. It's been well established that changes in these hormone levels affect many processes such as growth and development, metabolism, and even heart rate. T3 levels have also been shown to have effects on the nervous system. Most notably are its effects on sleep behaviors. However, the mechanism behind this neuronal activity is still not completely clear. Our lab hypothesizes that the reason for this activity is that T3 works similar to substances referred to as neurosteroids. No, not that kind of steroid. These compounds work by interacting with neurotransmitter-gated ion channels, which is exactly what our GABA-A receptor is. Neurosteroids mostly work on these type of receptors as allosteric modulators. This means they bind in sites on the receptor other than the active sites, such as where GABA binds. The neurosteroids then act to either enhance or inhibit the activity of the ligands binding to the receptor. Our lab group hypothesizes T3 works in this way, similar to a neurosteroid, due to the structure of the molecule. Looking at other types of known neurosteroids, there are many similar structural groups to that of T3. Working on the premise that T3 works as a neurosteroid, our lab decided to perform molecular simulations of T3 interacting with the GABA-A receptor to see whether or not it would bind to the receptor and what kind of effect T3 would have on the receptor. We first started by aligning the T3 molecule into a site we believe to be the allosteric binding site for neurosteroids in the GABA-A receptor. We hypothesized this is the site for neurosteroids because the crystallized structure of the chloride channel that we base our simulations on contains a molecule called ivermectin at this site. Ivermectin is a drug that structural groups and function are similar to that of neurosteroids. Once T3 is aligned, we place the pair into a bilipid membrane and surround it with water and ions. This way our simulations mimic the interactions that would be present if it were in an actual cell. In this graphical representation, you can see the membrane in a beige color, but you cannot see the water and ions. Here you can see all the atoms in the system, but it's difficult to see the protein, so we will go back to the previous representation. Because the binding site is close to the membrane, we will cut the portion of the receptor above the binding site and then look from the top down. Here we can see how the T3 is interacting with the receptor and how the parts of the subunits behave. One interaction we are interested in is between T3 and the serine residues of the receptor displayed in yellow. This is because we believe there is hydrogen bonding between these two, which may cause important changes in the receptor. You may notice that the membrane is not fully surrounding the receptor. This brings us to the first step of our simulations, the minimization step. At this step, we put restrictions on the receptor in T3 and allow everything else to sort of settle in around them. After this, we take off the restrictions and allow everything to run for about 200 nanoseconds of time. This may seem small, but in a cell these interactions work through small time frames. Pausing here, we begin to see when the T3 molecules embed itself deeper into the receptor. There is also a T3 molecule that begins to leave the receptor. In the rest of the movie, this T3 molecule will come back into the receptor, and another T3 molecule will begin to embed itself. Let's replay the movie without any interruptions. We believe that T3 molecules bind well with the GABA receptor because the molecules embed themselves into the receptor and interact with the serine residues. Our future studies will focus on the binding affinity of T3 for the GABA receptor to further test our hypothesis. Thank you for watching.